So our topic for today is the Lac Operon. Now, the Lac Operon is a part of DNA which codes for a unit of gene expression in a bacterium. So let this be our part of DNA, that is the Lac Operon. Now, this Lac Operon will consist of two parts. One will be the regulatory gene, and the other will be the structural gene. And there will be one more part in between. We'll come back to it later. Now, a structural gene is a gene which codes for proteins required within the cell. And the regulatory gene produces repressor proteins, which control the function of a structural gene. So let's elaborate on these aspects. So our structural gene will consist of three parts in the Lac Operon. This is the Lac C, Lac Y, and Lac A. Now this Lac C will code for beta galactosidase. This Lac Y will code for beta galactosidase permease. And this last part will code for the enzyme trans-acetylase. These three will together form our structural gene. Now, what does beta galactosidase do? So this beta galactosidase will always slowly convert lactose to allolactose. Secondly, it will also digest lactose into glucose and galactose. We'll come back to the function of these molecules later. Now, this permease is an enzyme which is present in the cell membrane of the bacterium. This allows lactose to enter a bacterial cell only in absence of glucose. Now, once this lactose slowly enters the inside of a bacterium cell, the beta galactosidase will convert the small amount of beta galactosidase present will convert our lactose into allolactose. Now this allolactose will begin its function. So to understand this, we need to come back to the regulatory part of our Lac Operon. So let me draw this out again. So the regulatory gene of a Lac Operon produces or codes for a repressor protein. Which is something like this, suppose. After our regulatory gene, we have the promoter region, the operator region, and then our structural gene. So in absence of allolactose, this repressor protein will bound to our operator site, the operator region of our Lac Operon. Now, once this happens, RNA polymerase can no longer transcribe the structural portion of our Lac Operon. Hence, the 3 enzymes coded for our structural gene is not produced. But when our allolactose is present, let me draw this out. Now, when allolactose is present, this will go on and bind to a binding site in a repressor protein. Now, our repressor protein is what we call allosteric. That is, it has 2 binding sites, one for allolactose and one for the operator region of our DNA. So when allolactose binds to our repressor protein on one site, the other site, which usually binds to the operator region, is blocked. So when this happens, the repressor protein gets shifted out. Of the operator region. And now, as this is now open, our RNA polymerase can bind to the promoter region and transcribe Lac Z, Lac Y, and Lac A to produce their respective enzymes. Now, the action of permease produced by Lac Y has already been discussed, but the action of beta-galactosidase is still left to be done. So one function of this was to convert lactose into allolactose, which acted as an inducer, which in turn shifted our repressor protein from the operator region. But beta-galactosidase also digests lactose to glucose plus galactose. This is in turn used in respiration. And once our lactose is broken, less allolactose can be formed. This in turn frees our repressor protein, which in turn switches off the structural gene. Along with this, as we now have glucose present, this will also block our permease enzyme. And if our permease enzyme is blocked, then lactose can no longer enter inside the bacterium. Now this has a significance because in presence of glucose we don't actually need to respire lactose anymore. Now there is only one more thing left to be discussed, that is our beta galactosidase also converts our galactose molecules or cleaves our galactose molecules to form toxic compounds. This is where our Lac A comes into action. Now Lac A was this part of the Lac Operon, and it codes for the enzyme trans-acetylase. This trans-acetylase adds an acetyl group to our galactose. And this acetylation prevents it from being cleaved by beta-galactosidase. Hence, toxic compounds do not build up within the bacterium. So that's about it. Hope you guys understood the concepts, and if there is any way in which I can improve my explanations, please note them down in the comments. See you guys next time.